Kia ora koutou. My name is Mick Black. I am the co-director of Genomics Aotearoa, and it is my pleasure today to be hosting the Genomics Aotearoa seminar for today. Today, I am beaming in from my temporary office, which is gradually filling with junk while we have our lab renovated. It is my great pleasure today to welcome along Laura Duntsch from the University of Auckland. So Laura is coming to us after completing, at one point in her career, a BSc in biology back in Germany, an MSc in animal ecology in Sweden, and then a PhD in conservation genomics at the University of Auckland. Since then, she's been working as a postdoctoral scientist at LIC in Hamilton, but today she is going to be talking to us about conservation genomics and the Hihi, I can never pronounce that, a threatened Aotearoa New Zealand Passerine. Jump in, Laura, and correct me, and then take it away.
It is Passerine, but we accept both.
Welcome along. Thank you very much.
Thank you. I'm gonna start sharing, and maybe you can give me a quick yes if you can see it.
Yes.
Excellent. Tēnā koutou katoa, ko Laura tuku ingoa no hamene o ko tipuna kei te no au ki Kirikiriroa. Thank you for the introduction, Mick, and welcome everyone. My name is Laura. As Mick said, I'm currently a postdoc at LIC, but today I will be taking you on a virtual nature walk to the island of Tiritiri Mātangi in the Hauraki Gulf and present results from my PhD thesis that was entitled Genomic Insights into the Adaptive Potential of the Hihi, a Threatened Aotearoa New Zealand Bird. I already now want to quickly also take the opportunity for to thank the various funders and supporters of this project, as well as my PI Anna and those that provided the images that you're going to see throughout the presentation. So let's start with a bit of background on Hihi. Hihi are a small forest-dwelling endemic species only found in New Zealand, and they were once common across the North Island of New Zealand. But human interference, such as deforestation and the introduction of mammalian pests, have led to a decline in Hihi populations until they were extinct from the mainland in the late 19th century. The island, here displayed in orange on the map, sorry the dot is a bit small, was the only remnant population for Hihi until the end of the last century, really, when they were then gradually reintroduced to different sanctuaries around the North Island. The population my study individuals are from is from Tiritiri Mātangi, which is the topmost black dot here on the map. But let's zoom in. So it's up there in the north in the Hauraki Gulf. It's about 1.5 hours with the ferry from the Auckland CBD, which made it very convenient to reach for me as I did my PhD up in Auckland. Yes, and it's on this island where I spent a few months during my PhD as a doc volunteer and helping others collect the data and just helping, maintaining the conservation work around Hihi on the island. The data that I am using for my own project was already collected before I started my PhD. And some stats about the island itself. So it's 220 hectares large. And in the last century, the island was basically stripped of nearly all its native bush following human occupation. It was long used for cattle grazing, but thanks to dedicated restoration efforts, approximately 6% of the island is covered in native bush today. And together with that, also, the mammalian predators have been eradicated, and the island is now home to not just Hihi, but many other native birds, reptiles, and invertebrates. And like the bellbirds are in the thousands. The project is managed by the Department of Conservation, but obviously in partnership with the local iwi, Ngāti Manuhiri, and the supporters of Tiritiri Mātangi. But yeah, the star of today's talk is, of course, the Hihi. There's about 190 on the island at the moment. I hope you can hear me over the sound. But yeah, you see here the Hihi on the in one of the sugar feeders. There's about 6 of them across the island. So Hihi almost exclusively breed in nest boxes, and they have this sugary water here available all year round. And as you can see, they have to share it with bellbirds because they're about the same size, so both of them fit through the mesh. But yeah, over the past 30 years or so, many conservation and volunteer efforts have led to, not just a thriving Hihi population on the island, but also the establishment of quite a unique long-term study system. And as a result, researchers have been able to investigate some important questions around Hihi behavior and phylogeny and population dynamics, and also translocation management practices. On a genetic level, there were a few microsatellite studies that estimated inbreeding depression and assessed genetic diversity in Hihi, and one more recent study that has also been diving into the question of Hihi adaptive potential, which is where my thesis kind of sits in the grid as well. So what kind of data do we actually have available for Hihi? Hihi are definitely a unique study system to work with. In the early stages, there was an observed pedigree for the population on the island. And then in the early 2000s, microsatellite data helped verify this pedigree. And ever since, every bird born on the island is genotyped and placed in the pedigree based on that data, and sexed as well. So that's, for more than about 20 years now. There's also been the previous PhD student in my lab. She has been designing a SNP array, and for that there was 10 Hihi that were red sequenced and also had whole genome sequencing data available. And during my PhD time now, I was using this SNP chip data, where theoretically it was a 40K chip for 1,500 birds. So that was not just on Tiritiri, but also on for some of the other populations across the North Island. But Tiritiri by far had the highest individual number on there. In reality, I only worked with about 500 of those birds, but I will explain a bit later why that is. And recently, complementing the kind of genomic toolbox for Hihi has been the release or the publication of the male and female Hihi genome assembly by Sarah Bailey et al. Earlier this year. So why is this data so cool to have? Well, species which exist in fragmented and reduced populations can suffer from quite a range of genetic impacts. For example, populations often lose significant amounts of genetic diversity as they reduce in size, which means they are potentially losing important adaptive diversity that enables them to respond to current and future environmental changes. At the same time, random genetic drift becomes stronger relative also to natural selection. And then there's also inbreeding. So my thesis aimed to learn more about some of these aspects for Hihi and make sure that I am using the right tools. So the story of my thesis kind of wrapped around two main gaps. First, there was the thought that there's still a bit of a lack of insights from genomic approaches applied to natural populations. And the Hihi study system kind of presented an ideal opportunity to change that. And secondly, even though there's extensive information on Hihi morphological and life history traits, actually there has been only very little genomic work done to this date that I started my thesis. So genomic work is definitely crucial to uncover potential evolutionary threats to the species and complement ongoing conservation management. So the aim of my thesis was to explore and evaluate modern molecular tools and approaches in a conservation context, while at the same time shed some more light on the adaptive potential of this unique species that is of conservation concern. So for this talk, I'm narrowing it down to 3 projects of mine, so in total, I published 4 chapters within my thesis, and I'll be talking about 3 of them. One is about a sample mix-up that has happened. The second one about comparing data of different resolutions to estimate inbreeding. And then the last one is to actually measure inbreeding in a group of Hihi. So let's get started. As I mentioned, just before I started my PhD, a SNP array has been designed to genotype lots of Hihi. However, Anna detected numerous incorrect genotype ID associations, and before I started even my PhD, and concluded that a sample mix-up must have happened that basically affected all Hihi samples within each population, which was quite dramatic. And it came, of course, as a bit of a shock, but also changed the mission for my very first data chapter, which was now focused about resolving these sample mix-ups. However, first, I conducted a survey because we were thinking that we can't be the only ones that this happened to. So I surveyed the research community and found that almost 80% of responding researchers have encountered sample mix-ups in their lab, and got nearly 300 responses, which was great. However, in my subsequent literature review, I also noticed that many recent studies do not appear to systematically report those sample checks as part of their publications, which makes it difficult to assess whether sample mix-ups have occurred or have been detected or performed at all, really. So yeah, there's apparently lack of a kind of standardized sample verification framework prompted me to establish a sample check framework myself. So based on the survey responses and what tools we had available, we worked on a framework that could help perform sample checks for data such as ours. So here's a picture of the framework as it appears in our publication. It utilizes existing metadata such as species and population structure and sex and pedigree information to perform sample checks. And yes, I am aware that all study setups differ. Hence, I would like to point out also that some of these steps can really be performed with only very little metadata available. Lastly, I also want to demonstrate its application to our very own Hihi data set. So we had this puzzle of who's who and kind of could confirm at least 488 individuals, so about 39% of all genotyped individuals, helped us determine who they are and could use them for subsequent work. And overall, I was very appreciative of how honest some of the survey responders were about sample mix-ups. And I've heard a lot of wild stories. And was just really hoping that this chapter kind of emphasizes the value of routinely checking genetic and genomic data for their accuracy. So I guess the main message is check your samples carefully. This is my take home. And this here's the title of my paper in case you would like to read up on it more. That was kind of journey one, or part one of my Hihi journey, my PhD. So now we had the individuals that we wanted to work with, and we knew that we wanted to estimate inbreeding for a larger group further down the way. But how accurate are SNP chips actually compared to other data sets? That was the question that we asked ourselves. So in this part of my thesis, we tried to shed some more light on this question by performing an in-depth inbreeding analysis based on runs of homozygosity. And for that, we used the GA reference male Hihi and 10 Hihi, for which we had RED-seq and whole genome sequencing data available because they were used to design the SNP chip. So there are many different inbreeding coefficients, of course, but whenever I from now on mention inbreeding, I mean inbreeding based on runs of homozygosity. How inbreeding generates runs of homozygosity can be seen here in the schematic, where segments of the genome of the bottom individuals are traced back to one of two ancestors. So the orange descends from the male and the blue from the female. So we see that recommendation creates some mixture between those chromosomes in the first generation. However, the breeding of these two related individuals generates an offspring in the third generation, which has a large overlapping tract of blue segments. And this overlapping identical by descent segment is called a run of homozygosity. So when you sum up all these segments, usually above a certain threshold size, and divide them by the genome size, then you get an inbreeding coefficient called FROH, and it ranges between 0 and 1, and the higher the value, the more inbred the individual is. Anyways, so we first looked at variants from a reference genome male to investigate homozygosity landscapes and inbreeding levels derived from datasets that have different densities and distributions, but stem from the same individual. So the genotype, we called this male yellow, by the way. So when I mention yellow, that's that reference male. So the genotype calls were obtained by mapping the long reads back to the reference assembly and then calling the sequence variants. And then we ended up with basically, in this case, I'm presenting 4 different data sets, the first one being the full genome one, and then 3 data sets that resemble the data we had for the other 10 birds in the study. So the kind of magnitude of whole genome sequencing data, SNP array data, and red-seq data. So the exact identical positions, basically. So we used this data for 2 different processes. First, we used a really novel approach to see what our software, in this case we used Flink, could detect based on the number and distribution of sites that we have in our data set. So that's called the genome coverage parameter. So basically it's the maximal RH length the analysis can possibly discover. And that's calculated by converting every single SNP in the data set to homozygous, and then determining the total inferred size of homozygous regions, and then dividing this number by the total assembled genome size. So you can see here that the genome coverage parameter for the different data sets ranged from 54% for the red-seq data to 93% for the SNP array data and 98% for the whole genome sequencing data. So the take home from this was that SNP chips, offer a fairly high genome coverage, which is good to know. And I just also want to mention that this is a subsampling approach that is basically applicable to all species and can help anyone assess the power of their data sets to accurately infer inbreeding levels before even starting any analysis. In addition, we assess the concordance between the full genome and each of the other data sets by subsampling the full genome to the SNPs shared with the focal data set and then calculating the proportion of SNPs called within a run in the full genome that were also called within a run in the focal data set, in the same time for basically the exclusion of a SNP. And the main takeaway here is that despite a relatively low SNP density, the SNP array data successfully called 81% of true runs and correctly identified 88% of regions that did not contain one when comparing to the full genome output, which was very promising because it was kind of similar to or not too different from the whole genome sequencing data, whereas there's quite a bit of a drop when looking at rat sequencing data resolutions. When looking at specific contexts and their run of homozygosity landscape for the different data sets, we find that low density data sets tend to miss runs of homozygosity in some places and overestimate their length in others, which results in quite contrasting homozygosity landscapes. As you can see here in those plots, those are 2 different contexts, and then basically the display of the runs for each of those contexts per data set. However, we also saw that, you can see that here on the left-hand side, that these sort of patterns were not necessarily reflected in the overall inbreeding scores, which were fairly similar to each other. And the main takeaway here was that, that SNP array data can yield comparable inbreeding results to full genome densities and performs a bit better than red-seq data. And lastly, we extended our analysis to the 10 Hihi, where we had low coverage whole genome resequencing, red-seq, and SNP array genotypes available, mostly to evaluate the relative effects of sequencing depth versus SNP density and missingness. And again, we see quite a high correlation between what was in our study our gold standard data set in the SNP array data, but the red sequencing density did not seem to correlate very well. We found that high rates of missingness, especially downwardly bias both the number and length of runs of homozygosity found. But on the bright side, when removing individuals that had low coverage or very high missingness, those correlations usually improved. However, with the SNP chip data, we don't really have those problems, I guess, which is another reason why we thought we could trust it. So yeah, the main takeaway from this was that when using genomic data to evaluate inbreeding, studies definitely have to consider that the estimates of those runs of homozygosity are really dependent on the analysis parameters, the density of the data set in the sequencing depth. But we also saw that runs of homozygosity from SNP chips are fairly concordant with whole genome sequencing data and definitely perform better than just a few thousand red-seq markers. I also want to point out that in this analysis here, we used the software Plink, but when we submitted this paper to a journal, the reviewer asked about the software RZooROH, which is an R package. In comparison, and that one uses a likelihood-based method to estimate those runs instead of Plink using a rule-based method. So we did both in this paper, and you can find those results in this paper here if you're interested. So yeah, now we have the situation that we have a good enough data set, and we have a bit more confirmation that our SNP chip data will be able to come close, at least, to the true inbreeding values and patterns. So yeah, now let's apply this knowledge and add to the Hihi genomics toolbox by measuring inbreeding and inbreeding depression. A first dip into Hihi genomic properties and topics such as Hihi adaptive potential was already done by Pierre in 2019. Where he used reduced representation genome sequencing as well as long-term phenotypic and fitness data from the Tiritiri Mātangi population to find quite a lack of molecular genetic diversity at a genome-wide level, low heritability of traits under selection, and fairly low additive genetic variance of fitness. And in combination, said that these results support a lack of adaptive potential in this threatened species. Now I wanted to build on top of those findings using more samples and high-quality SNP data and change the angle slightly to focus on one of those other threats to small populations that I mentioned earlier, inbreeding and inbreeding depression. So we did this analysis for 363 birds from the island. The reason why it's a bit less than what we had initially is because they had to have remained on the island for their entire lives, and many birds throughout their lives are being translocated to other populations. So yeah, firstly, we wanted to explore genomic inbreeding patterns, both total inbreeding, but also the timing of inbreeding. Then we wanted to measure genomic inbreeding effects on fitness. And finally, we used a genome-wide association approach to test the local specific effects of inbreeding on fitness. And we're actually one of the first ones to do so. We used analysis based on a Zoe sheep publication by Martin Stoffel et al. And he also very greatly helped me with our own Hihi model. So I'm very grateful for that. If you're interested to learn more, here's a link also to my thesis. Where you can find all the chapters. And for this part of the analysis, make sure you use the updated female GA Hihi genome and updated our SNP chip positions accordingly. So yeah, our paper in this case summarizes 4 main findings from our run of homozygosity investigation of Hihi on Tiritiri Mātangi. With the first one being that some individuals show much higher than average inbreeding levels. You here see the histogram with the inbreeding values at the bottom, and then the count of how many birds have that specific inbreeding value on the y-axis. So inbreeding in this case and for this chapter was actually measured with the software I mentioned earlier, the RZOO package in R, because it simply better accounts for allele frequencies and genotyping errors. The cool thing about runs of homozygosity is that we can actually use the relationship of recombination and the length of the run to track the demographic history of a given population. So those that are only very recently inbred are expected to have significantly longer runs, which cover a greater expanse of the genome, since, recombination hasn't had the time yet to break it up. And the program I just mentioned is very useful in a sense that it helps to identify which homozygous segment dates back how many generations, so the different colors. So if we look at the 10 most and least inbred birds and the timing of inbreeding events here in this plot, so you have the birds on the x-axis and the full inbreeding level on the y-axis, and the different segments dating back to different inbreeding events. So we can see that although all individuals have similar levels of ancient inbreeding, which is here in orange and red, highly inbred individuals are affected by recent inbreeding, which is the blue colors, which we think can likely be explained by bottleneck effects, such as the habitat lost after European arrival, but also their translocation to the island in the 1990s, which is roughly those 5 generations ago, as the light blue color indicates. And thirdly, we also wanted to look at the effects of inbreeding on various fitness traits, as we have all this information for Hihi. And our group, at least, was spanning about 10 cohorts, so 10 breeding years. So the fitness traits we were looking at were lifetime reproductive success, annual reproductive success, juvenile survival, and adult annual survival. So we tested the effects of inbreeding, but also the effects of sex, lifespan, and age on any of these fitness traits. What we found was that the global inbreeding significantly affected juvenile survival, but none of the remaining fitness traits, as you can see here on the left. So the fixed effects that are on the left are the only ones that were significantly affecting the fitness traits on the right. So inbreeding affected juvenile survival, and there was also a sex effect. So females were a little bit more likely affected than males. But this kind of, to us, suggested that inbred hihi are removed from the population before they even reproduce, and that there are hence slight indications for inbreeding depression in hihi. Now, the last thing we did in this chapter was this more novel approach that I mentioned earlier, which was a genome-wide association study. So normally, those are used to identify markers that explain differences in gene expression or phenotypes such as height. But here we performed a GWAS to see whether there's an association of any of the homozygous SNPs that are in a run of homozygosity with one of our 4 fitness traits. So here's a Manhattan plot, as many of you are surely used to. At the x-axis, you see the different chromosomes, and the y-axis displays the associated p-value. And if the dots are red, that means they're above a significance threshold and are significantly associated with a or negatively associated, but significantly associated with the specific trait. In this case, lifetime reproductive success. So you can see we were able to identify 13 SNPs that were significantly associated with this trait. However, they all had fairly small effects, and we could not find any of these sort of signatures for any of the other fitness traits that we looked at. So the findings in this last thesis chapter of mine suggest that inbreeding depression does impact hihi, but at different genomic scales for different traits, and that purging has failed to remove all variants with deleterious effects from this population of conservation concern, and that, as always, more data can definitely shed more light on it, especially higher density data can look into more detail into those regions specifically. And yes, again, a screenshot just of the paper, which is also open access, that was published earlier this year, if you're interested in reading more about it. But yeah, in conclusion, my thesis was able to add a little bit to the Hihi toolbox by moving from genetic to genomic data analysis for the first time, at least for such a large group of Hihi. And even though my findings kind of support, again, the theory that adaptive potential is limited in Hihi, I hope that studies such as mine can still assist in active species management, even if they just encourage to continue long-term monitoring. And of course, Hihi work didn't stop with me finishing my PhD. The wheel keeps turning, and there's now new students taking up new work. And some of the future projects are, for example, to perform whole genome resequencing data for 250 Hihi across different cohorts, and then look at things such as structural variants and immune genes, the role of purging in different genetic loads in the different populations. And yeah, I mean, species such as Hihi will benefit from these advances. Hopefully they will inform recovery strategies and population modeling and future translocations at some point. Something I definitely noticed throughout my PhD is that there's not really a one-size-fits-all approach when it comes to evaluating a species' evolutionary capacity, but rather many different non-genomic and genomic approaches that will probably continue to complement each other. And yeah, they will come with their advantages and disadvantages. But yeah, hopefully they will teach us about a species and their capacity to adapt to future change. And so at the slide, just to say that Hihi also sometimes make the news, because they're quirky and a bit different, and they are also sometimes used for large studies that use worldwide big data sets, because they're so special and they have so much data available. They're an awesome resource also for studies from abroad. And I also want to use this opportunity, even though it's still, I think, about 2 months away, to plug for Hihi for the Bird of the Year, or in this case, Bird of the Century election that is coming up, I think, in November. I mean, I think all that you've heard is even more reason to vote for Hihi. So I'll leave that there. And of course, it takes a team to do all this work. It's not possible to do any of this without the help and support of many people. So my lab group has been amazing. Anna, of course, a stellar supervisor. My advisors, Patricia and John, that are based in London at the moment. And then my lab group that, I mean, I've done most of this work during COVID times, so we've probably had as many in-person meetings as we had Zoom meetings. So it's always good to see other faces and have some uplifting conversations. And so thank you very much. I think some of them are here today as well. So thanks again. And with that, I'm done, and happy to take any questions, or if you want to email me later, if that's what you prefer, that's totally cool as well. Thanks.